Hey everyone, we're going to go ahead and go over Unit 6, which was the Mendelian and non-Mendelian genetics before your test. I'm going to go ahead and go through just a couple of key points in your review like I have been doing, and then go through a couple other things that are important to help you be successful. Last test before I come back and before the semester ends, so make sure that you're doing everything that you can to do well on that as well, okay? So starting in your review, there was really only two standards. The first one is making sure that you understand Mendel's laws. So there's a lot of key vocabulary that came with Mendel. So remember, he did his P-plane experiment. He gave us terms like dominant and recessive, homozygous, heterozygous. We want to know what those things are. So the first thing during his experiment, Mendel, he's the father of genetics, is the first person to realize that we get two copies of each gene. Remember, those are the alleles that we get from each parent. So parents are each contributing 50% of their DNA down to the offspring in the form of an allele from each copy. Remember that the vocabulary words allele means it's a version of a gene. So it's kind of like a flavor, so to speak, of the trait. So if the gene is for eye color, the alleles could be for blue eyes or brown eyes or green eyes or so on, okay? Those alleles combinations can be in a couple of different ways depending on what they inherit from each parent. So remember, if it has dominant and a dominant, it's going to show dominant. Remember, if it's recessive and recessive, it'll display recessive. But if it has the dominant and a recessive trait, we call that heterozygous, and it'll always display the dominant. If it's Mendelian genetics, that's also known as complete dominance. Okay, that was his law of dominance. So you also want to know his two separate laws here, law of segregation. When you hear the word segregation, that should kind of mean separate to you. This is really just showing what you do on a Punnett square. You are separating the gametes or what's inside the gametes for the parents, and that way only one allele gets passed down to the offspring. So when the baby's gametes are formed, that's what's showing. They've separated into separate ones. And then the law of independent assortment. So independent should mean it's kind of doing it on its own. And assortment is giving you a variety. So we see this with those dihybrid Punnett squares, where we're getting different combinations of traits being inherited. So maybe it's all of mom's features or all of dad's features, even though you're still getting DNA from both parents. Or maybe you get a different combination. Okay, so the alleles of different genes separate independently of one another. So for example, if we're looking at maybe hair color and texture, maybe I inherited my mother's hair color, but my dad's hair texture. Or maybe I got both of mom's hair color and texture. Or maybe I got something completely different that wasn't present before. So they're all separate traits, okay? And then in addition to just Mendelian inheritance, we're looking at just complete dominance and recessiveness. We have some non-Mendelian complex patterns as well. So before Mendel, we didn't really know much about inheritance. And then after him, we've learned a lot more with technology and further experimentation. So we do know now that there's a lot of other complex patterns in nature. So those are those things that you've learned about, like multiple alleles, where you have more than just a dominant and a recessive allele. You can have many. Okay, like blood type, for example, there was 3. You have an A, a B, and an O allele. You can have codominance, where both phenotypes show. You can have incomplete, where both phenotypes kind of blend in and give you something different. You can have polygenic traits, where you have multiple genes working together. You can have sex-linked traits. So those are specific on the X or Y chromosome. In our case, it's only going to be on the X chromosome. So these are all a little bit different. So make sure you know those vocabulary. It's going to be really important that you know the vocabulary for this unit to be successful. So utilize your Quizlet, the flashcards, do the little tools, the little true and false, or the little games that they have on Quizlet to help you practice. Okay. So let's go ahead and look at a couple other things. All right. So again, with just Punnett squares, your whole test is mostly Punnett squares. If you're not doing Punnett squares on your answer sheet, so don't write on the test, but you can use your scratch paper or an answer sheet, whatever the sub gives you to utilize, make sure you're doing Punnett squares, okay? So one parent's always on the top, the other parent's on the side. It doesn't really matter which one is which unless it specifies. Remember, this is that law of segregation here where the gametes are going to separate, or the alleles are going to separate into each separate gamete. When they come together here, that's showing that process of making the gametes or meiosis. So this would be what's inside of the offspring. These are all the potential combinations, so to speak. Remember, Punnett squares show the probability. Okay, so we'll talk about analyzing that in just a moment. Some basic vocabulary. Some of you are mixing this stuff up when you check your mini quiz for the unit. So you have to know genotype versus phenotype. Genotype in this case is the combination of genes or alleles. Okay, so those are the letters. Phenotype, think of the PH as, think of it sounds like physical. So phenotype is the physical trait. What color is it? Is it tall? Is it short? Is it what eye color? What hair texture? Does it have freckles? Those are all phenotypes, okay? If you do not know the difference between homozygous, heterozygous, dominant, and recessive, you're not going to do well. So you have to know what those look like. Okay, remember we express dominant alleles with capital letters. We do recessive with lowercase. That way it kind of shows you that if we have a dominant and recessive together, dominant is going to be what shows. Homozygous, that prefix homo means same. So homozygous dominant is going to have two capital or two of the same uppercase B's. Homozygous recessive, it's going to be two of the same lowercase alleles or letters, two lowercase b's. Heterozygous, that prefix hetero means different. So you're going to have two different alleles. In this case, one dominant and one recessive. So it looks like one capital, one lowercase. Make sure you're reading test questions carefully. Make sure you read them more than once. If it tells you that the parent is homozygous and their parent's heterozygous, you want to make sure that you're writing the right thing when you're doing your Punnett squares. So again, vocabulary is important, but also making sure you're reading the test questions is really important. Okay. When you analyze them, you're expected to know or be able to analyze a Punnett square and be able to give a ratio, a percentage, or a fraction. Most of you are pretty good on percentages, but making sure that you can translate that into a fraction or a ratio. So if we use this example here where both parents are heterozygous, we get these potential offspring here. So if we analyze the genotypes to get a ratio or a fraction or a percent, we can see we have one homozygous dominant, we have two heterozygous, and one homozygous recessive, which if it asks for the genotypic ratio would be a 1 to 2 to 1. You always go in the order from dominant to recessive. If it asked about any of these in particular in terms of fraction, 1/4 would be here for the homozygous dominant. 1/4 would be the homozygous recessive. 1/2 is 2/4 simplifies to 1/2 for the heterozygous. Or if we did them in percentages, 25%, 50%, or 25%. If we analyze this and then ask for a phenotypic analysis, remember that is looking at what trait. So in this case, dominant showed red, and the phenotype for recessive was white. So the phenotypic ratio, notice it's different from the genotype, is a 3-to-1 ratio, where 3 of the offspring will be red, 1 will be white. Or again, fractions 3/4 versus 1/4, or 75% or 25%. So make sure that you know how to translate the ratio to the fraction to the percent. Make sure you know the difference between a genotypic analysis versus the phenotypic analysis. You guys also learn about dihybrid crosses. So remember, this is looking at two traits. This is where Mendel's law of independent assortment comes in, because here we can have peas that are yellow and round, or green and round, or green and wrinkled, or yellow and wrinkled. So we're getting new combinations that we didn't have before. Those traits are inherited separately. They don't go together. Okay. So these are a little more complex because you're looking at two traits. So you have to know what this FOIL method is. Okay. So if these were the parents' genotypes, notice there's 4 letters and not 2 because there's 2 traits. You have to FOIL these to figure out the gametes that go on the top and on the side. So FOIL stands for first, outside, inside, last. If you look at the arrows here, this kind of tells you how they got this on the top and the side. You want to do it in order. So here the first goes with the first. So this big R and big Y, which is why I put that here. In this case, both parents are the same. So we did it for the first box in either one. Outside, do the two outside ones. So that's this big R with this little Y. Okay. We have our inside and then our last. Okay. That's what goes on the top and on the sides. Once you do that, you just fill it in the same way that you did normal Punnett squares, but you want to format things a couple particular ways. So you keep the letters together. So you see R stay together, Y stay together. You still write the capital letter of that pair before the lowercase. So big R comes before the little r. Okay. And then you would analyze it the same way. Okay. These ones are just a bigger Punnett square. So just take a little bit more time. Okay. And then the last thing you guys did was go over a bunch of different complex patterns of inheritance. I'm going to go back to your review to go over some of those examples. So you guys had these different pictures here. So again, make sure you know the definitions. So in this first example here, where you crossed a red and a white, notice both genotypes were capital letters. They all came to be this hybrid here, which was in a capital R, capital W, and they were all pink. So crossing a red and white where you're getting this blending in would be incomplete. So if I was there, I would have taught you guys think blending in, incomplete. Okay. So if you had a blue and a yellow organism and it displayed incomplete, you should expect green. If you had blue and red and it blended in, you should get purple. Okay. If you look at B here, if you look a little closely, so it says the female gametes, it has these X chromosomes and the males has an X and a Y. Notice there's nothing on the Y chromosome. You should have learned that the Y chromosome is smaller, so it doesn't carry all of these traits like we do on the X. So that's going to be X-linked or sex-linked. Okay. With sex-linked traits, males are typically more effective because they only have one X chromosome. They don't have a second one. So percentage-wise, they're more likely to have it. With sex-linked traits, females can be carriers, males cannot, because they could be heterozygous, males cannot be heterozygous. Males inherit their sex-linked traits from their mothers because that's who they get their X chromosome from. Okay, so just a couple things there. Sex-linked traits, you can analyze them a little bit more. So you can look at just the female offspring versus the male offspring, which you can't necessarily do in a regular Punnett square. So it may ask you questions like, of the male offspring, how many have the sex-linked trait? And in that case, it would be out of half, okay, instead of 4. It's not asking total offspring. So again, be careful with how you're reading the questions for those types of sex-linked traits, okay? For C here, we have multiple alleles because we can see here we have 3 different alleles, which is why we write them a little bit differently with the little superscript there. So we have A, which is dominant, and B, which is dominant. So that's with the capital I and the capital A, or the capital I and capital B. And then we have O, which is recessive, which is why it's a lowercase I. It's the only recessive trait, or allele, excuse me, so we don't have to put the O on top of it, okay? But for all of these, because there's more possible genotypes, we can have more possible phenotypes, okay? So we have a homozygous version here for type A, IA, or we have the heterozygous version, which is the capital IA and the lowercase I. It's still going to show type A because remember, O is recessive, okay? Same thing for B. And then AB is actually a special thing. It's called codominance, like you've learned about, because they're both equally dominant. It'll be both A and B blood, okay? These Punnett squares are the same thing like this. They're just formatted differently, okay? So again, make sure you know the vocabulary. If it tells you it's a homozygous type A, you'd want to pick this genotype versus the other one, okay? Here you have an example of polygenic traits. So skin color, height, eye color, those are all examples of polygenic traits. Polygenic is going to refer to anything where you have a wide variety. So with skin color, people are just not white or black. You have a lot of different shades in between, a lot of different shades of white or tan or black. So Same thing with eye color, not just blue eyes or brown eyes. There's different shades of all of these. So that's because there's multiple, poly means multiple genes working together. So you can see here, these genotypes are really long. These are 6 letters long or 6 alleles long. You don't need to know how to do Punnett scores for any of those, but know those examples: skin color, height, eye color for polygenic. And then E here, of course, is codominant. If we look at the example here, these are different cows. We have a red cow and a white cow, and gives us this kind of spotted cow. So if I were there, I would have told you to think of they both show. So in codominance, both of the parents' phenotypes show up in the offspring, typically as some type of spots or stripes, but not always. So this is like speckled chickens, the AB blood type, the spotted cow in this case, okay? So make sure you know all of those as well. And then lastly, you learned about some pedigree. So you want to know the different symbols. You want to know squares are males, circles are females. Shaded has the trait, half shaded could be a carrier, okay? But these last two are really important. So these last two questions are looking at how do you know if it's a sex-linked or autosomal trait, or how do you know if it's dominant or recessive? So when you took some PowerPoint notes, you should have learned that if it never skips a generation, it's dominant. If it skips a generation, we can probably assume it's recessive, but that's not always the case. But dominant traits never skip a generation. So if we look at this first one, we see it's in generation 1, 2, and 3. So we know it has to be dominant. But if we look over here in question 25, we see it skipped generation 2. So we can probably assume that is a recessive trait. Autosomal versus sex-linked. Autosomal is pretty much every trait that you learned about except for the sex-linked traits. There's not a lot of sex-linked traits. Sex-linked traits are things like color blindness, hemophilia, okay? So in this first one here, we want to look at the number of males versus females that are affected for autosomal or sex-linked. So I can see here we have 3 females or 3 circles shaded. We have 3 squares or 3 males shaded. So because it's about equal, or in this case it is equal, we assume it's autosomal, okay? If maybe it was even like 4 males and 2 females, we would still say it's autosomal. But if we look down here at 25, we only see that it's 3 males that are affected. So because only males are affected, we know that males are more affected by sex-linked traits. We assume it's sex-linked, okay? So you want to be able to identify those different patterns as well, okay? I hope you guys go through and study what you need to. You have your review packet, you have your assignments, you have your vocab flashcards, you have your Quizlets to do electronic games with. You have the Kahoot that I'm putting up for you guys, this video. So do what you need to, rewatch any Amoeba Sisters that you need to. Good luck, and I can't wait to see you guys soon. Okay, take care.